Hi everyone, my name is Stephanie Ciarlone and I'm in clinical development at Ptc Therapeutics. First, I wanted to say thank you so much to Paula, Allison, everyone at FAST, and all involved in working so hard to organize this virtual meeting during these unique times, and for allowing us to share our update. It's truly a pleasure to be able to share with you an overview of PTC and our Angelman syndrome program updates. So who is PTC? PTC is a global, commercial, diversified biopharmaceutical company focused on innovative therapies for rare genetic disorders. PTC was founded in 1998, so we're a little over 20 years old, in South Plainfield, New Jersey, with a focus on developing drugs for rare disorders. Since then, we have established and grown our presence there, maintaining our corporate headquarters and research laboratories in South Plainfield. Our international headquarters is based in Dublin, and we have medical marketing teams based in Zug, Switzerland, and our LATAM headquarters is in Sao Paulo, Brazil. So we do have global commercial capabilities as well as infrastructure. In just the past 2 years, we've added offices in Cambridge with the acquisition of Agilis, of which many of you are well aware, and also in California through the acquisition of BioElectron, as well as a new biologics manufacturing facility in Hopewell, New Jersey. We are now a global company with a footprint in over 50 countries, PTC offices in 20 countries, and over 850 employees. Ultimately, we are passionate about our purpose. Our mission is to bring the best-in-class therapies with clinical benefit to patients affected by rare disorders. We see ourselves as more than just a biotech company. We are a cause. At our core, we are scientists and have always been driven by the science. We are innovators who find new ways to treat devastating rare diseases. But for us, it's more than finding treatments. We are committed to patients in the rare disease community. At our core, PTC is an innovative company focused on applying scientific expertise to bring novel therapies to patients with rare disorders in need. Stuart Peltz, our CEO and founder, has a background as both a scientist and a professor. Many of the platforms and therapies you see here were actually internally discovered and developed, while others were brought in to complement our deep scientific roots. As you'll notice, we have a robust pipeline across all stages of development, including multiple late-stage products and an expanding research portfolio. We continue to build our pipeline of potential medicines by analyzing our scientists' research and collaborating with other groups. We also use a multi-platform approach to drive scientific breakthroughs and are actively working to develop treatments for multiple rare disorders and rare cancers, further extending our deep and diverse pipeline. In 2018, we expanded and diversified our pipeline even more with 4 gene therapy programs. Overall, as you can see here, PTC is a committed player in CNS indications, and this breadth demonstrates both commitment and experience. In August of last year, we acquired a state-of-the-art biologics facility in Hopewell, New Jersey to support gene therapy production for Friedreich's ataxia, Angelman syndrome, and beyond, bringing full manufacturing capabilities in-house. We felt that controlling our fate with manufacturing is key to our strategy, and this means not only having the necessary facilities in place, but also the essential talent. This facility in Hopewell, New Jersey will help PTC provide the best products for clinical trials and human use. Our approach in gene therapy is differentiated, with a focus on treating rare monogenic disorders, primarily in the CNS and retina, which correlate with low turnover cells. With AADC deficiency, we have submitted the MAA in Europe for permission to bring our therapeutic to market, and we expect the EMA's Committee for Medicinal Products for Human Use's final opinion for the AAD deficiency application in the first half of 2021, and we now anticipate the BLA to be submitted in the first half of 2021 as well. For our FA and Angelman syndrome IND-enabling activities, they are progressing well. And lastly, we are working on multiple exciting non-clinical development candidates. Now to talk more specifically about gene therapy and Angelman syndrome. Gene therapy is the process of delivering the missing gene, in this case UBE3A, directly to the appropriate cell types using an engineered viral vector. There are many considerations for any viral vector, and specifically for UBE3A delivery. This includes which vector type. AAVs or modified AAVs are most commonly used for their overall safety. AAV serotype is another consideration. Is it 2, 5, 9? Which will provide us with the best transduction and expression in our targeted cells? Then there are promoters, which drives transgene expression, or the amount of UBE3A. Which isoform to use There are 3 known isoforms of UBE3A, likely with varying localization, which is best to include. And finally, route of administration, which impacts transduction patterns, or where you will see the gene expressed. All of these make up the AAV vector components and are significant considerations. I know most of you have seen this work before, but for those of you that haven't, this publication was from Dr. Weaver's laboratory at USF back in 2011. This was the first in vivo proof of concept work that demonstrated increased UBE3 expression in the AS mouse with AAV9 and murine or mouse UBE3A. Injections were directed into the hippocampus bilaterally, meaning on both sides, and expression levels were normalized to wild type. They also reported corresponding improvements in hippocampus-associated behavioral deficits, like fear conditioning or water maze, as well as LTP, or long-term potentiation. This is also known as synaptic plasticity, which ultimately controls how neurons communicate with one another. More recent work in collaboration with USF demonstrated increased UBE3 expression in both the AS mouse and rat models following a different route of administration, ICV injection, which is intracerebroventricular or into the lateral ventricles in the brain, directly into the CSF. This modified AAV not only demonstrated increased UBE3 expression overall, but also showed a greater expression when compared to AAV5. Additionally, this AAV showed increased expression in the CSF when compared to AS non-treated animals, as well as AAV5 treated rats. While this isn't necessarily the exact AAV we're proposing to use in the clinic, this demonstrates that tweaking or modifying various AAVs can lead to increased expression and potential improvements. Even more recent work has been completed internally by PTC examining various routes of administration and continuing to work to identify the best and most optimized vector. The work I'm showing here portrays a snapshot of some of this positive data in both AS mice and rats, which again does not necessarily represent the exact AAV we propose to use in the clinic. This includes increased UBE3 expression in various regions throughout the rodent brain, as indicated by the purple staining in the panels in the top left. Typically, in an AS animal, you will see little to no staining of UBE3A, and the image you would see would look almost black versus being brightly lit. These images are all from AS-treated rodents and demonstrates relatively robust expression. Additionally, we have been able to demonstrate improvements in synaptic plasticity, as was seen in Dr. Weaver's prior work in 2011 that I mentioned earlier. We not only view these improvements in AS mice, but in AS rats as well. Finally, we have seen phenotypic improvements in things like the Hyman-Classman phenotype over time in AS-treated animals, which is an indication of improvement in general neurologic function. Overall, we have seen increased UBE3 in the brain, leading to both molecular and phenotypic improvements in either AS mice or rats, which is very promising. Finally, I'd like to provide you with a status update. Where are we today? We've taken part in the ABOM steering committee to stay as up to date as possible with the latest thinking related to the direction of ideal biomarkers and outcome measures in Angelman syndrome. Overall, we're working as quickly and safely as possible to develop the necessary animal studies to support first-in-human clinical trials. We're also working to gain insights and learn as much as we can from previous and ongoing AS natural history data to assist in the overall design of our clinical development program. Finally, as I mentioned previously, manufacturing is critical, especially in gene therapy. Therefore, the acquisition of a state-of-the-art biologics facility will help to establish the manufacturing process internally to provide the best products for clinical trials and human use. So I know a lot of parents ask, what can I do to help? Other than good record keeping, including the timing of tests completed by our child's physicians, having robust data in things like the Global Angelman Registry and natural history studies would be helpful. Participating in things such as this would allow us and others to access this data to help us learn more about Angelman syndrome specifically and potentially help inform and assist in the design of successful clinical trials. And that's it for my talk. Thank you so much again for having me and allowing me to share our program update.